As I mentioned before, my name is Gabriel Gasque, and I am Head of Outreach at Protocols.io. Welcome everybody to these tips and tricks webinar for Protocols.io power users. And before we begin with the material, let me tell you a little bit about myself. I have a background in experimental neuroscience with doctoral studies and postdoctoral training. And before joining Protocols.io as their head of outreach, I was an editor in PLOS Biology, which is a flagship journal of the Public Library of Science, and I was there for 9 years. I consider myself a scientist, as well as an advocate for open science, experimental reproducibility, and research integrity. I am recording this session, and I will share the recording with all registered participants. So these are the topics that we will be covering today. So I'll show you how to bring into the platform existing protocols in different formats. We will talk about the file manager, which is a tool to organize, archive, display, and audit your protocols. And we will also talk about the components of the editor, some of them. Regarding the file manager, we'll talk about the difference between copying a protocol or forking a protocol, as well as creating new versions. I will also mention how to create collections. And finally, I'll talk about sharing of protocols. This is not an introduction to Protocols.io. This is an advanced webinar and presumes some basic knowledge of the platform. In case you are not familiar at all with Protocols.io, I would invite you to visit the different resources that we have for training that you can see on the slide now. So we have regularly scheduled webinars like this one. This is an advanced one. But if you click the link, or if you follow the link, or if you go to protocols.io/webinars, you will see that we have several topics. So we have introductions, the philosophy for getting, and the strategies for getting credit and recognitions by using protocols.io. And you can choose different time zones to register. And so this is the list of the upcoming webinars, and you can register. We also have at protocols.io/tutorials, pre-recorded tutorials that explain the tools and features of the platform. And if you scroll all the way down, you'll find a link to request a one-to-one demo. In these demos, you will talk to one of our specialists, and you can share your screen, they can share their screen and have a very interactive conversation about any question that you might have about the platform. But let's now jump into the live demo. So the first topic that I want to cover is how to bring into the platform existing protocols. So I'm gonna minimize my presentation here. And I'm gonna go to my workspace just to orient ourselves. So to the left, here in the platform, we have all the workspaces to which I belong. You might have at least one, but you can have more than one if you belong to more of one workspace. So I have my workspaces here. We're gonna be using this one. Then this is the file manager for that workspace. Every workspace has its own file manager, but I'm gonna be working on this one, which is my private workspace. And these are the folders and documents within that workspace. And what I want to do is I want to bring into the platform existing protocols. So here I have my hard drive in my computer, and I have several protocols here in different formats. So I have a protocol that is a Word document. I have a protocol that is an Apple Pages, a PDF, and a plain text. Just to show you, I can open this protocol. So this is a protocol that, one of the protocols that I want to import into the platform. You know, it has a title, it has an abstract, some notes, and then the steps of the protocol start here, and the different steps are organized into different sections. I'm going to close it. So I'm going to show you 3 ways in which you can bring existing protocols into the platform, starting from the easiest and then going to the one that is more time-consuming, but also create a more sophisticated and useful protocol. So, but before I do that, I'm going to create a folder, a new folder, and it's going to be called May 8, 2023 demo. And this is where I'm going to be importing my folders, sorry, my protocols. So the easiest way to do that is just to, I can just take them and transfer them by dragging them into that folder. But I can also go to New, Upload Files, and I can browse my computer, or I can just drag and transfer them there. So I'm going to try to do that. And now the protocols are being copied. And let me see if I copy them here. No, sorry, it wasn't that folder. So this one. No, I copied them to a different location. Well, I'm going to do it again. I just can take them and drag them here. So now my 4 protocols have been transferred into the platform. The advantages of doing it this way is that it is extremely simple, as you have just witnessed. I have also centralized all my protocols in a single space, which is my devoted workspace for protocol development. And I also can share this protocol with anybody within and outside my institution by just typing their email. So I will show you very quickly, even though we will touch on sharing again at the end of my presentation. So I can share this with anybody just by typing their email address. And then, and I save it, and I shared that document with my collaborator. Now, a limitation of doing it this way is that I cannot publish these documents because they are not protocols. They are these documents in their existing format, in their pre-existing format. So they are still a PDF, a Word, a Pages, or a text. So I can share, but I cannot publish. I'm going to show you a very quick way that takes just 2 seconds more to create a publishable protocol out of these pre-existing documents. And to do that, I'm going to create a new protocol. I'm going to save it in this folder that I just created today. I'm going to just choose the biology template. Now, one of the advantages of creating this protocol is that I can add a title and an abstract. And by adding a title and an abstract, I will make my protocol more discoverable because it will be searchable if I ever publish it. So I'm going to call it May 8th demo 1. I will do not create steps. At this point, I can add an abstract here. This is a mock abstract for demo 1. And the way I'm going to bring the protocol, my document, my pre-existing document into this protocol is by attaching it. So I can scroll all the way down in the description tab, click attachment, and I can select it from the file manager, or again, I can just drop it. Because I have imported my documents already, I can go to the file manager, to my folder, and I can select the protocol that I want to attach, and I have attached my protocol. So if I go to view, this is how this protocol would look like. So it has a title, it has an abstract, and the protocol itself is the attachment in a PDF format. So again, I have centralized the protocol. I can share it the same way that I showed you before for that existing document, but now I can publish this one because this is a protocol. It doesn't have steps. The protocol is an attachment. And I'm going to show you the third way, which takes a little bit more time to, I'm going to rename it just to avoid the typos. So I'm going to create a second protocol, but in this case, rather than attaching the document, I'm going to import the steps of the document into the steps of the protocol. So I'm going to create a new protocol. I'm going to call it, I'm going to save it in the same folder. And I'm going to call it May 8. Again, I can add May 8, demo 2. I can add in the description tab, I can add another abstract here. And then what I'm going to do, going back to the steps, and this only works for text files. So you can't do that. You cannot do that if your protocols are in a PDF. But if they are in a pages or in a text file, you can do it. So I'm going to open my pages, which is equivalent of Word for Macintosh. So here you can see, as I showed you before, the protocol. And I don't know if it's too small for you to see, but it says here, if you have an existing protocol, you can insert steps from a text file. So I'm going to click it. I'm going to go back to my protocol, select the steps that I want to insert, which are these two that belong to the first section. I'm going to paste that text here. Each number is a step. I preview. And now the steps in the text document have been transformed into the steps of the protocol. I can even copy the name of the section and paste it here. And I can do it again for the next section. So I can copy the 4 following steps and paste them here. Preview, add to the protocol. But now these 4 steps, from step 3 to step 6, they belong to a different section. So I'm going to create a new section by selecting this, set new section. And then I can copy the title of the section. And replace it here. Right? And now I have created two sections. Each of them has been coded in a different color. And if I go to view, again, you can see that it has a title. It has a mock abstract here. And then it has the 6 steps organized in 2 sections. There is a table of content. Now, the advantage of doing it this way is that in addition to centralize the protocol and make it shareable and publishable, now it has become an interactive and dynamic document. So it will accept comments. So if I publish it or if I share it with anybody, that person or those readers can add comments. And if I post a comment, if a reader or a collaborator posts a comment, they will be notified, or you will be notified, the author will be notified. So you can see that one comment was added. And I can also run that protocol, this protocol in the platform. And I'll show you how to do that in a bit. But before that, I want to go back to edit. And I want to show you some useful components. So to reach out to the components, I can open this tool here that will bring me to the components. And a nice component here is the timer. So as you can see, some of the steps are timed. So there is a 10-minute incubation. Here's a 1-hour incubation, and I can transform these into a duration component, which will create an actual timer. So a 10-minute timer there, a 1-hour timer here. Right? I can also transform these into concentrations. These are percentages. So I can go to concentration and say this is mass percentage, for example. Oh, no, sorry. I made a mistake there. I wanted to mark this one, the 65%. And this is a 65 volume to volume. I can do here a 29 volume to volume and keep going. So one advantage of doing it this way is that when I go to view, all these times and concentrations become more visible. And particularly with the timer, if I decide to run this protocol in my computer or tablet to generate an experiment record, when I run my experiment, as I finish each step, I can mark it as done. And then when I reach the step that has a timer, I can actually click the timer and it will start counting down. And when the time is over, my computer or my cell phone will beep, letting me know. And you can set different timers for different times for different steps. So I'm going to go now back to the file manager. I'm going to reload. So you can see that I have created my 2 protocols, May 8 Demo 1 and May 8 Demo 2. And I have shown you 3 ways in which you can bring protocols into the platform. So let's keep working on editing this protocol. I want to show you now how to insert tables, embed videos, and 2 components, what we call the step case and the smart component. So inserting images or pictures is extremely easy. I select the step in which I want to insert an image, and I just click here, insert image or video. And I can browse in my documents the image that I want to insert. And it's done, right? And I can even add a text to my image here. In this step, what I'm going to do is I'm going to embed a video that I previously found in YouTube that shows how to load a gel. So to do that, I'm going to select embed media, but first I need to find that video on YouTube. So I have my video here on YouTube. You can see it. It shows you how to create and load a gel. And to insert this video into my protocol, I have to go to share, select the code for embedding, copy it. I go back to my protocol, and I select here embed media. I can paste the code and save it. So now the video has been embedded. So I don't see the link. What I see is actually the video. And if I go to view my protocol, I can see the picture that I inserted in the first step, and then the video that I embedded in the second step, and I can actually play that video in my protocol. Okay. I'm going to go back to edit. And now I'm going to show you how to insert and create tables. So I'm going to create a table in my second step, in my third step. So this is a tool to create tables. I have created a table here, and I can say here, this is the reagent. I can say volume. And 10x. So this is reagent A, B, C, and I can add my volumes here. And then here, I can add my formula, which is going to be B2 times 10. And then I just drag this. And now I have multiplied column B by 10. I can add a legend to my table, but I can also bring a table that I have pre-created in Excel. I'm going to show you how to do it here. So I have here my table in Excel with reagents, amounts, and 10x. I can copy my table. Sorry, I'm going to close this one. And I'm going to insert here my table and just paste inside the table the content that I just copied. And you can see now that I have inserted my table. Now, there's a trick here because in this table, 10x is actually a formula that you can see here. But the formula was not transferred to my protocol. So here I just have the values. I don't have the formulas. If I want to transfer the formulas, what I have to do in Excel, I have to select View, show formulas. And now I can see the formulas. I can copy this again and paste it here. And now the formulas have been copied. Not just the values, but the formulas. And I can edit them, right? Let's say I don't want this to be 10, I want this to be 15. So I go here and I can edit my formula and just drag it. I want to show you another component that is called the smart component. The smart component looks like a table. So I have to open the components, insert the components, and select the smart component. And the smart component is something that I will show you. For example, it's an interactive component that is very useful when you are using protocols.io to run your own experiments. And I want to show you, for example, if you are counting cells in a field, in a microscope field, and you want to count the number of dead cells or the number of stained cells. So the key is going to be total cells, and you can make them a requirement. And then in a second row, I'm going to put stained cells. And I'm also going to make it a requirement. And add the component so it looks like a table. And this is very useful when you are running your experiment. So I'm going to go again to view. So now you can see the protocol with all the tables and figures and videos. And here's the smart component. So if I run that experiment, as I proceed, let's say I'm done with step 1, I'm done with step 2, 3, 4. If I reach step 6, because I made this component compulsory, I cannot mark it as done until I enter the values for this particular experiment. So let's say that I counted 39 total cells, of which 24 were stained. Now I can mark it as done. And if I save the record of the experiment, those values will be saved in my record. So I finish the run. And then if I go to the file manager, I will see that there is a record of that experiment. And if I open that record, those values will be saved. So you can run your experiment, you can run your protocol in the computer and save those specific values for that specific experiment on that day. I'm going to reopen my protocol to keep editing it. And the last thing that I want to show you is what we call the step case. Some protocols might need a bifurcation for 2 different conditions. Let's say that you usually work with 2 different tissues, or you work with 2 different bacterial strains, and the protocol is the same up to a point, and then depending on the tissue or depending on the bacteria, you bifurcate the protocol. And this is the symbol for the step case. So I'm going to add it here. Or here, new, step case. So let's say that I work with heart. And so if you are working with cardiac tissue, do this. And I can add the step case, and then I can add another. And let's say I'm working with liver. And then here is if you are working with liver, do that. And I can add the step case. And now my protocol can alternate between heart and liver. If I go to view, so again, I can view my protocol. And then in the last step, I can choose between keep working with heart and or changing to liver. So this is a bifurcation, and you can keep adding more cases. So it's not only two. Now I can say lung. And then I have, so my protocol is the same from step 1 to step 5. And then in step 6, depending on my tissue, I can have different steps. So this is all I want to show you about the editor. There are other components and other tools, but the best way to learn about them is by just playing around. But these are probably the most tricky components that might take you a little bit of time to figure out on your own, and that's why I'm presenting them. Please, if you have any questions, type them in the chat or in the Q&A section, and I will get to them at the end of my presentation. I want to go back to the file manager and explain to you the difference between copying a protocol, forking a protocol, or creating a new version. So as you can see here, I can copy or fork, but I can only copy my own protocols. I cannot copy protocols that were published by someone else or created by someone else. I can only copy my protocols. If I create a copy of my protocols, these two copies will be independent from each other. So we recommend that you use copies when you are, for example, considering putting together many protocols that are independent from each other, but they all belong to the same working group. So you have the same authors and the same funders. So you share the metadata, but the protocols themselves are independent from each other. So you can create a template with the authors, the metadata, and then you make several copies that then you customize further. But the protocols are independent from each other. If I fork a protocol, there will be a historical link between the original version and the forked version. And forking usually works when the protocols are somehow related. Let's say I create an original protocol that has been optimized to work in mouse, but now I want to adapt the protocol to work in rats. And I have to make some changes for the protocols because rats are larger and the tissue might need more digesting time. So I have my original protocol, and then I can fork it. So if I make a copy, as I said before, the protocols will be independent from each other. But if I make a fork, and I'm going to call it B, there will be a historical link between the two protocols. And I can see that here. So this is version B, and the platform says that it was forked from an original protocol. And I can actually go to that original protocol by clicking this link. So that's the difference between copying and forking. I can also create a new version. So by creating a new version, the protocol will be linked in a more tighter way than with fork. And I want to use version when I want to make a correction to my protocol, for example. Let's say I created a protocol and I'm optimizing it with very small changes, or I publish my protocol and I realize that I make a mistake. Published protocols are not editable because they receive a DOI. So if I publish a protocol and I want to make a correction, I will need to create a new version. And by creating a new version, the protocols will be linked to each other very tightly, more tightly than the fork. Now, what about public protocols? So I'm going to search for a public protocol. Oh, but to do that, let me change to Safari, because in this account, it's my work account, and I have like super user privileges, but I want to show you in this, which is my civilian account. So what about public protocols? So in principle, I can use any public protocol as a template to optimize it for my own conditions. So I'm going to search the platform for protocols, public protocols. In this case, this protocol has 3 versions. And this is how they will be linked to each other if you create versions of your own protocols. And you can navigate to the different versions of the protocol. But if I find this protocol and I think, oh, this is a very useful protocol and I want to use it for my own purposes, but I need to make some changes because I'm working with a different equipment or a different cell line. So I can make an editable, this protocol cannot be edited because it has been published and has received a DOI. But I can make my own copy or fork. Now, because I didn't create this protocol, because this protocol was created by somebody else, I cannot copy it. I can only fork it. And in this way, the history of the protocol will remain. So I can make a fork of that published protocol. I'm going to select in what folder to save it. And now, that copy is my own fork of that public protocol. And it has the flag that I showed you before, where it says that this protocol, my own version, has been forked from this original one. And now I can go back and edit this protocol to fit it to my own needs. I can even change the name and say, this is my own version, and I can edit it as I want. So if I go back to my file manager, I can see that I have created this new fork. It has my name, and it has an indication that it was forked from an original one. So I have explained the difference between copying, forking, and versioning, and when and for what purposes you will do one of those. And now the final thing that I want to show you in the last minutes is how to share protocols, how to get a link that you can include in a paper, how to publish, and how to invite members to your workspace. So again, let's work from this. My personal workspace. If I want to invite a collaborator to join my workspace, I go to the administration menu. I select manage members. And then I can invite people to join my workspace. And I can do it by typing their email address. And then add them. And then once I invite people, they will appear here. And if they accept the invitation, they will appear here. So that's the way to invite people to the workspace. You can also make your workspace fully public, meaning that it will be searchable. So if people go to this and type the name of your workspace, they will find it. Or you can make it private, and this is the default setting. And if you make it public, you can select whether you want it to be open to all, so anyone can join your workspace, by request only. So there will a button will appear that says request to join, or by invitation only. So I'm going to go back to my private. I'm going to go back to my working space. So if I search here for COVID, for example, and then I search for workspaces, I can see all workspaces that are public that have COVID as a keyword. And for example, in this case, this is a public workspace where I can see the publications associated with the workspace, the members, but I cannot join this workspace because it has been set to by invitation only. But there are other workspaces that might be fully open. So this again is a public workspace where I can see the members, I can see the publications, but if I click join, I will be automatically joining the workspace. And you can control that for your own workspace the way that I just showed you. Now I'm going to go back to the file manager. And sorry here. And the last thing that I want to show you is how to share, publish, and create a collection. So to share, I can take any protocol or any document. And if I don't want to bring people into my workspace, once I bring people into my workspace, they will have access to all the documents that are stored in the file manager of that workspace. But if I want to share just one protocol with somebody outside my workspace, I select that protocol, go to share, and can send that protocol by using the email address of my collaborator, add it, and then I can control what that collaborator can do with this protocol. They can just view it, edit and publish or co-admin that specific protocol. And if I say, okay, this person can view and edit, and if I save it, then this collaborator will receive a notification. I can also get a link, which this private link, you can copy it and you can share it via Slack or WhatsApp or email and send it. And if the person follows this link, they will go to a view-only version of your protocol. So they will be able to see it, but they will not be able to edit. And this is a good link if you want to include this protocol in a submission of a paper that's being peer-reviewed. And you can even get a blind link in which all affiliations and authorship will be removed from the protocol. And to publish it, I can publish any protocol by selecting publish. And I just have to complete 6 easy steps that include keywords and abstract and funders. In addition to publishing, which will make it public, permanent, and assign a DOI, I can also just reserve a DOI. By reserving a DOI, I will make my protocol permanent with a DOI, but it will not be made public until you finish publication. So if you want to fix the protocol and receive a DOI, to have a DOI to include in a submission, for example, of a paper, you can do this, but the protocol will not be made public until, for example, your paper has been accepted, and then you can finalize the publication. And it will be made public with the same DOI that you reserved. And now the very last thing that I want to show you is how to make a collection. You can put together many protocols in a single collection. So I'm going to make a collection for today. To make a collection of the protocol, I can select new collection. I'm going to save it in today's folder. And I'm going to include I'm going to search just in my files, my protocols that I created today. So I can select demo 1, demo 2, and the protocol that I forked. But I can also include in my collection public protocols. So I can include a public protocol. And now if I go to view, I have created my collection. Let me go back to edit so I can add a title. I'm going to call this May 8 collection 1. Now I can go to view and I can see my collection has 4 protocols, which I can open each of those. And now this protocol has an additional flag that says that it's in one collection. And that collection is a collection that I created today. And this collection, I can also publish it as a single document with all these protocols included. So if I publish a collection, it will receive a DOI. So for example, if you are sending a paper with many protocols, you can create a single collection with all the protocols that belong to that publication and get a single DOI for that whole collection. And with that, I'm going to stop. I share a lot of information, and I will be happy to take questions. So somebody is asking me to demo the sample component. So let me go to I'm going to open this protocol. And I'm going to add a new step and the sample. So here it's you add the name of the sample and the description of your sample. For example, could be a diseased tissue. And I can add the description. So this tissue was removed from patient 1. And that's the sample component. And if I didn't answer your question, let me know and I'll give it another try. So somebody else is asking what protocol data and metadata can be used to search for protocols. So we search for title, we can search for authors, you can search for affiliation, you can search for keywords, and you can search for reagents. Let me show you. So here, I can search for Drosophila, and it will search in the abstract, in the title, and in the steps for the keyword Drosophila. If I wanted to make it an exact search, I have to use quotation marks. So for example, I can search for single cell PCR, and it will search for all protocols that have single cell or PCR. But if I do quotation, it will search for the exact match. And there are none. Because the exact match doesn't work. But now it is exact match for single cell if you put it in quotations. I can also search for protocols for author or institution. So that's some of the metadata that can be used to search for protocols. Let me know, Marlena, if I am answering your question. And you are asking if you can edit reagents once they are created. To be honest, let me see if we can do that. So let me go back to editing. I'm going to insert a reagent here in a step 6. Where is reagent? Reagent. May 8 demo reagent. Oh, I'm going to create a new reagent. May 8 demo. And save. New England Biolabs. And then if I go to materials, no, I don't think that you can edit it, but let me double check, Marlena, and get back to you. Would you, it is my impression that you cannot edit it. But if you type your email address, I'll get back to you. Or you can send me your email address via email, and I'll get back to you. But I don't think that you can edit it from what I can see now. Okay, I have your email address now. There are other questions in the Q&A. Oh no, these are the questions. Anything else? Oh, sorry, you're asking another question which I missed. So can I share only a folder created in my workspace with collaborators? Yes, well, you cannot share a folder. I think you can share a protocol. Let me go to the file manager. Oh, I think you can share a folder. Let me see. Yes, you can share a folder. And if you share the folder, you will not be sharing anything else in the workspace. So if you share a protocol or you share a folder, you are only sharing that folder or that protocol. You are not bringing people to the workspace, and they will not have access to any other document that is in that workspace. And as I showed you before, you can control what these people can do with what you are sharing. You can make them only viewers or viewers and editors, or viewers and editors and publishers. Did I answer your question, Marlena? So if I share my folder, and I add, you can control what this collaborator can do with those documents. View, edit, and publish, or you can make them co-admin. But even if you make them co-admin, they won't have access to anything else that is in the workspace, only with this very specific protocol or this very specific folder that you are sharing with them. Oh, and Beth is asking about what are the export options. So if you want to export, so you can export to the printer as a PDF. You can export it as JSON. And if you select PDF, you can export to your computer, which is what I'm going to show you now. So no, that's not what I wanted. Export. Sorry, let me take this one. Export PDF and to the computer. Okay. So I'm exporting it as a PDF. And, oh, because this is the collection. I chose the one. This is the best one. So I'm going to export this one as a PDF to my computer. So I am exporting this protocol, which I created previously, as a PDF to my computer. And I can also print it, or I can download it. Right? So I created a PDF to be exported to my computer, but I can also export it to any of these cloud-based drives. I can send it to Box, to Google Drive, to Dropbox, and we also have integration with Lab Archives, which is an electronic lab book. Did I answer your question, Beth? So we are now over time. I don't want to take any more of your time. If you have any questions, drop me an email at protocols.io at, oh, it's not here, sorry, at Gabriel at protocols.io, and I will get back to you. And I will get back to you, Marlena, regarding whether a reagent that has been created is editable. Thank you so much for coming. Thank you so much for all your questions and engagement. And I hope that the presentation was useful and that protocols.io is useful for your research.